So in this particular case, I wanted to just illustrate what the results actually look like and how you can see the percentage of heteroplasmy. So in this particular patient, you can look, this is the control sample, and you can see that we're looking for this G13513A mutation. So we're going to be looking right here at the G that's in the middle of this gray shaded box. And you can see that the peak heights are almost equal in the control patient that has that normal G allele at this particular position. In your patient, you can see that in blood, the peak height of this G is lower and the height of the A is higher, because what's happened is a percentage of these cells have the A mutation here. So remember, 2 A's in a row is going to give you a double peak height. And so in this particular case, this would be an example of what the data would look like if the patient had about 30% of the mutant A allele and about more than 70% of the normal G. And then down here in muscle, you can actually see that the percentage of heteroplasmy is higher. So here you hardly see any of the G peak, and you see that this A peak is very high. And so in this case, in the muscle cells, the patient actually has 64% of the mutant allele and only 36% of the normal G nucleotide at that particular position. Traditional PCR and sequencing technologies typically require about 20% heteroplasmy to be able to reliably detect the mutation. And this is all sort of theoretical. It varies based on the mutation and some other factors. But with pyrosequencing, you can theoretically detect heteroplasmy as low as a few percent. So in addition to it being quantitative, it's also more sensitive. And this can, I think, really be of value when you're looking at relatives for carrier testing who may have a very low percentage of the mutant allele, and it may be that their percentage of heteroplasmy for the mutation is so low that it would be missed by traditional sequencing, but it might be able to be detected by pyrosequencing. So I'd like to talk now a little bit about next generation sequencing. You may have heard this terminology. This is kind of a popular phrase in the research community and starting to become more common in the clinical diagnostic laboratories. There are 3 main next generation sequencing platforms. They each have their unique advantages and disadvantages, and I'm not going to talk in specific detail about them, but just in general, next generation sequencing, regardless of which platform you're using, is high speed, high throughput. So the sequencing process is basically occurring simultaneously on millions of different DNA templates generated from your genes of interest. And so you're able to sequence multiple genes, so essentially hundreds of exons at the same time. I'm going to show you an example of what the data looks like in just a minute, but basically it generates what are called reads, which are short short amounts of sequence information. They often are about 35 base pairs long. You may produce up to 150 million reads with a single run. And so these reads then need to be aligned to your consensus or your normal wild type DNA sequence to be analyzed. So here would be an example of what this would look like. And so at the top, this line here would be the consensus DNA sequence. So this is normal. This is what you're comparing it to. Each of these short sequences here are reads, and so the computer program has very nicely aligned these for us, and you can see I cut this picture off. We had actually hundreds of these, but it would have been way too small for you to see. So you can see that we have many different reads for each particular DNA base that we want to look at. Here would be a location of a mutation. So you can see at this position, we would typically expect an A. And in red, you're seeing places where we're identifying a T. So in essence, about half of these reads are showing a T at this particular position. So this would be a patient who is heterozygous for a mutation. If the patient was homozygous for the mutation at this position, essentially all of these reads would be red. It is possible with this technology, because you're getting multiple different reads for each particular base, to pick up mosaicism that you wouldn't be able to see with traditional sequencing, and we have seen that at GeneDx. I don't think at this point anyone knows what the sensitivity is of that, so it's not something that anyone's going to advertise. We can't tell you, oh, mosaicism of 20% or more would be identified. We just don't have that kind of information yet, but we can tell you that we have been able to pick it up. So the advantages of next-generation sequencing are obviously that you can sequence hundreds of amplicons, so hundreds of exons at the same time. This gives you the most rapid turnaround time and is, at this point, the most cost-effective technology for large-scale sequencing. It's highly sensitive, so unlike the other two technologies, which really aren't good at picking up frameshift mutations, with this you're able to identify missense, nonsense, splice site, and frameshift mutations. And you get many different overlapping reads for each particular DNA base. So there are obviously advantages to that we've already talked about. But another one is that with traditional sequencing, you get one read essentially for each base. And so if your sequence didn't work, you have to go back and repeat the entire thing over again. And in this case, if one of those reads doesn't look good, you just don't use it because you have many, many others for that particular position. The disadvantages are really in the bioinformatics area. So as you can imagine, this generates an enormous amount of data, actually several gigabytes for each particular run that you do. So this requires aligning it and analyzing it and storing it, which I don't think I appreciated what a big undertaking that is until you think about if you're doing this in a clinical setting, you're obligated to keep that data for a period of at least 10 years. So we're generating gigabytes of data on every single patient, and then we need to be able to store that long term. So that is actually one of the biggest issues with trying to get this technology set up in a diagnostic lab. Right now, this process takes several days. So you put your reaction on to run, and depending on which technology you're using, it depends how long. But it may be 3 to 5 days until, from the time you set that sample up and put it on the machine until the time you've actually generated any data. So you hear people talk about sequencing the exome or sequencing the entire human genome, and really the limitation in a clinical setting is just that, if it's taking several days to get the data out, you obviously need a lot of different machines and they're expensive. So that's really one of the issues. And as this technology improves, we're going to see this process get faster, and that's going to take away a lot of those limitations. This also requires a critical amount of reads for each particular nucleotide position. So we usually like to see typically at least 20 reads for each nucleotide base. And if we don't have that for whatever reason, then we resort back to dideoxy sequencing to try to confirm the information. And abnormal results, again, should be confirmed with a different methodology.